Welcome to Fountain Education. Today we start a new topic, Basidomycetes, Basidomycota, large and diverse phylum of fungi, kingdom fungi, that includes jelly and shell fungi, mushrooms, puffballs, and stinkhorns, certain yeasts, and the rusts and smuts. Basidomycota are typically filamentous fungi composed of hyphae. Most species reproduce sexually with a club-shaped spore-bearing organ, basidium, that usually produces 4 sexual spores, basidiospores. Basidia are born on fruiting bodies, basidiocarps, which are large and conspicuous in all but the yeast, rust, and smuts. The common name bird's nest fungus includes species of the genera Crucibulum, Cythis, and Nidularia of the family Nidulariaceae, order Agaricales, which contains about 60 species. The hollow fruiting body resembles a nest containing eggs, peridioles. The peridioles carry the spores when they disperse at maturity. General characteristics: it is the most advanced and large group of fungi, which includes toadstools, brachyfungi, berry clubs, puffballs, stinkhorns, earth stars, birds' nest fungi, and jelly fungi. Majority of them are saprophytes, causing decay of litter, wood, or dung. Some cause serious wood decay in the form of rots. Some members, like Armillaria melli, are destructive parasites, destroying a wide range of woody and herbaceous plants. The commonly known rusts and smuts also belong to this group, which are restricted to live tissues of plants. Mycelium types and structure. Mycelium is very well developed. The hyphae are branched, septate, spreading on or in the substratum. In parasitic form, the hyphae may be intercellular with haustoria or intracellular. The cell wall is made up of chitin and glucan. The mycelium is in the form of a weft of anistomosing and interwoven hyphae. In some forms, hyphae aggregate together to form plectenchyma, exhibiting varying extents of differentiation from mycelial strands or rhizomorphs. The mycelium passes through 3 stages before the completion of life cycle. 1. Primary mycelium. It develops from the germination of basidiospores. The septa are simple cross walls. All the nuclei in the mycelium are identical or homokaryotic. Each segment of the mycelium is uninucleate and is monokaryotic. It represents the haplophase. It may multiply by conidia or sometimes by oidia. Secondary mycelium. The mycelium arises by dekaryoticization of cells of primary mycelium. The cells of the secondary mycelium are binucleate, and it represents a dekaryophase in the life cycle. There is increased prominence of dekaryophase in Basidomycotina compared to Ascomycotina, and this phase is independent, long-lived, and thus plays a prominent role in the life cycle. Dekaryoticization of the cells of the primary mycelium is achieved in different ways. Tertiary mycelium. This mycelium is also dekaryotic. The dekaryotic mycelium, which is involved in the formation of basidiocarps, is designated as tertiary mycelium. The specialized complex tissues of basidiocarp are made up of this type of mycelium.